Did you know investigators can use materials such as masks, clothing, cigarettes, glasses, or even tape to get DNA evidence? What exactly is the process of DNA evidence? Well, to help solve crimes, investigators look for DNA evidence where it shouldn't occur, and then they can test for a person to match. After finding a sample for a possible suspect at a crime scene, the investigators look for a known biological sample to compare the unknown sample to. While some jurisdictions routinely take DNA samples from arrestees during the booking process, sometimes a court order is required. Officials also take DNA samples from the victims to cross-reference against DNA found at the scene. The testing itself is done by professionals in a controlled lab setting. Most DNA samples submitted to laboratories undergo the following process. 1. Extraction is the process of releasing the DNA from the cell. 2. Quantification is the process of determining how much DNA you have. 3. Amplification is the process of producing multiple copies of DNA to characterize it. 4. Separation is the process of separating amplified DNA products to permit subsequent identification. 5. Analysis and interpretation is the process of quantitatively and qualitatively comparing DNA evidence samples to known DNA profiles. 6. Quality assurance is the process of reviewing analysis reports for technical accuracy. The DNA analysis process provides the analyst with a chart called an electrophoregram, which displays the genetic material present at each loci tested. In a complete profile, each person will exhibit either one or two peaks or alleles at each locus. In practice, evidence often contains a mixture of DNA from more than one person. These mixtures can be very challenging to analyze and interpret. If any locus is missing an allele, this is considered a partial profile. Partial profiles can happen for a variety of reasons, such as when a sample is degraded. If the sample has peaked at every locus, but any of them fall below a predetermined threshold, this would also be considered a partial profile. To enable profiles to be searched against a large national database, the FBI created the National DNA Index System, NDIS, in 1998. This national database is part of the Combined DNA Index System, CODIS, that enables law enforcement agencies throughout the nation to share and compare DNA profiles to help investigate cases. As of 2012, there are more than 10 million DNA profiles in the system, and CODIS has produced leads that have assisted in almost 170,000 investigations. Once a laboratory enters a case into CODIS, a weekly search is conducted of the DNA profiles in NDIS, and resulting matches are automatically returned to the laboratory that originally submitted the DNA profile.